Okay, so let's get started with our first speaker, Nabeel Qureshi from the US.
Good afternoon. My name is Nabeel Qureshi. So for the past 4 to 5 years, I've been investigating the role of a protein called telomerase in vascular function and investigating its emerging role in the mitochondria, all in the hopes of possibly developing a novel therapeutic to combat what's called vascular dysfunction. Now, the interesting thing about this project specifically is that it sits in the intersection between a variety of fields, such as cancer biology, cardiovascular disease, as well as the biology of aging. So before I started this project, I looked on Google and Googled what is the most prevalent cause of death in the world. Expecting to find cancer at the top of the list, I was very surprised by what I was able to find. I actually found that coronary artery disease was the leading cause of death in the world. Doing some more research, I also was able to find that coronary artery disease is an aging-related disease. So the more you age, the more susceptible you become to developing the illness. Additionally, I also found that another major medical dilemma was that dealing with the adverse effects of chemotherapy. So when a patient undergoes chemotherapy, that chemotherapy, while it is very efficient at targeting negative and malignant areas, it is extremely damaging to the cardiovascular environment specifically. So at the same time I was researching these two similar conditions, I was actually exposed to a unique enzyme called telomerase while I'm sitting in my high school biology class of all places. Now in high school biology, they teach that telomerase is characterized as an anti-aging related protein. So we have this anti-aging related protein, and we have this aging-related disease. I wondered whether the two could have any interplay. Could telomerase serve to mitigate the effects of both coronary artery disease as well as the adverse effects of chemotherapy on what's called the microvascular level? Now, the microvasculature is the smallest types of blood vessels in the human body, and they range from 50 to 150 nanometers in diameter, so they're very, very small. However, regardless of their small size, they're actually known to be highly important in cardiovascular integrity, and literature actually shows that they may be the first steps for the onset of major cardiovascular diseases, such as coronary artery disease, as well as atherosclerosis. One of the more seminal figures of my study, I'm able to show that given isolated human blood vessels, so those little microvessels, isolated from the time of surgery from both patients that have had coronary artery disease as well as chemotherapy for 20 to 30 plus years, just in a matter of 2 hours, by overexpressing this one protein, I'm able to make those blood vessels look physiologically healthy again. And I can explain why. So for the coronary artery disease, I'm able to show that the molecule required for the blood vessels to expand and constrict changes from a negative phenotype to a healthy phenotype when you overexpress this one protein. So in an older person or a diseased person, the molecule necessary to expand and constrict the blood vessels is a negatively charged molecule called hydrogen peroxide. However, as you overexpress telomerase, as well as develop a healthy phenotype, that molecule switches directly to nitric oxide, as you can see in the blue circled figure. However, most strikingly, in the chemotherapeutic pipeline, I'm able to show that if you have chemotherapy, most of your microvessels, or smallest types of blood vessels, actually do not constrict or dilate at all, given that they're almost dead. However, if you overexpress this one protein alongside these blood vessels, you can see normal vasodilation, so the blood vessels retain normal activity. However, if I was to go to a doctor today and say, okay, I'm going to give all your patients that have had CAD as well as chemotherapy this one protein, they would think I'm crazy. The reason why is that if you overexpress telomerase, conventional knowledge shows that you actually develop cancer. Because although you allow the cells to divide and divide and divide, that ultimately is what cancer is, a disease of the cell cycle, an inability for cells to arrest properly. So therefore, I wondered, could I create a mechanism in which I could preserve all these therapeutic effects, yet mitigate any of this resulting cancerous activity? And that's exactly what I was able to showcase in what I like to call my molecular Trojan horse. So in previous studies, which are not shown here, I'm able to show that telomerase actually has two localizations. It exists in the nucleus as well as in the mitochondria. I was able to show that the nucleus is responsible for that aging effect, allowing telomerase to extend the lifespan of these cells and possibly develop cancer at a later point. However, the mitochondria is responsible for all the therapeutic effects. So I developed what I like to call a molecular Trojan horse, which tricks telomerase for instead of going to the nucleus, it travels directly to the mitochondria. Therefore, I'm able to preserve all the therapeutic effects and mitigate any risk of developing cancer post-treatment. Therefore, making this treatment and drug delivery model is extremely clinically relevant. However, given the interdisciplinary nature of this project, I said, I just can't stop there, right? So what I did, given my interest in the mitochondria, I developed a full-scale mechanistic mathematical model of the mitochondria in both computational as well as mathematical language. So what you're seeing here is pretty much every single or a majority of the reactions of the mitochondria expressed in subenzymatic form. So as you know, an enzyme takes something from state A and goes directly to state B. However, in order to do that, that enzyme might take 20 to 30 individual steps, which in the literature previously has not really been mapped out formally. In my model, I sought out to do that. However, thinking more generally, this model not only is a way to simulate and develop more insights in relation to my project, but in theory is the most precise representation of the mitochondria to date, in that it accounts for subenzymatic activity and allows a plethora of mitochondrial components to be simulated computationally. So if you can take one thing away from my project, I think it's that in order to provide successful science, as well as science that's going to push the next frontier forward, it has to be interdisciplinary. In that, if you want to be a biologist, you shouldn't be afraid to pull concepts from chemistry, mathematics, physics, and computer science. Thank you.
Thank you very much. Okay, do we have some questions? Do you see a hand?
Yeah.
Ah, up there. Okay, perfect.
You said it was most of the reactions within a mitochondria.
Correct.
What are the exceptions, or why is it only most?
Gotcha. So as you know, the mitochondria is extremely complicated, right? So millions of reactions are going on in the mitochondria. So if you actually just take a look at one enzyme, where there may be thousands of these different enzymes, one enzyme might actually be 20 to 30 individual steps. So actually, what limits this model is what's been developed previously in the wet lab. So if theoretically, if every single reaction of the mitochondria was experimented on, maybe in prior literature, then yes, I could have included it in my model. However, given that it's only computational, I can't just create reactions that I don't know exist.
So that's basically all we know.
Exactly. So yeah, everything in literature formulated into a computational simulation.
Thank you.
Yep, no worries.
Is there any other question? Up there? Is this a question? Ah, okay, all right. Okay. Okay, then thank you very much.